Hello everyone, welcome to Get My Class. The question says chains A and B were combined to form human insulin by option A disulfide bonds, option B intrahydrogen bonds, option C covalent bonds, and option D both A and C. Now let us understand the question. It says chain A and B were combined to form human insulin by. So here we are talking about the formation of human insulin in vitro condition. Okay, in vitro by the method, that is by recombinant DNA method, okay, where they introduce the gene of there is gene for insulin into the cells, okay, into the cells, especially you can say the E. coli cells, okay. So the first they incorporate the gene for human insulin into the vectors and they introduce within the E. coli cells. And in the E. coli cells, so it is not that one particular E. coli cell will be able to produce the complete insulin. Okay. So what do they do? That human insulin consists of two main peptides. Okay, it is A peptide and B peptide. Okay. Then when this A peptide and B peptide combined by disulfide bonds, they combine by disulfide bond, then it will give rise to mature insulin. Mature insulin. Now, to produce insulin in case of or in vitro condition, what scientists have done, they have introduced gene for peptide A, gene for peptide A in particular group of bacteria, peptide A, and another group of bacteria they introduced. Okay, this is another another set of set where E. coli cells are there, but here the gene for peptide B have been introduced. Gene for peptide B. Okay. Now, what have they done? This group of organisms or this group of E. coli will produce peptide B chain. I'm telling. Okay, peptide B chain. Whereas this will give rise to peptide A chain. Okay, or you can say A peptide or B peptide. It is the same. You can just say A chain or a B chain. It's the same. So now this will be combined by disulfide bonds. Disulfide bonds. Okay, because the A chain, suppose this is the A chain and this is the B chain, these have to be, okay, to be functional, they have to combine with each other. They have to form bond with each other by disulfide bond. And now you have, this is A, this is B. And now what you have here is the mature insulin. Mature insulin, which has been produced in the lab condition. Okay. But here in natural condition, when it forms within the body, there will be another peptide which is called a C peptide. That will be your pro-insulin. That is, it is not the mature insulin. But that C peptide will be removed by the various process. And then the other two peptides, that is A peptide and B peptide, will combine together. And that will give rise to your mature insulin. So that happens naturally. But when it is done in the lab, this is how they do. They introduce individual gene, that is for A peptide separately and for B peptide separately into respective or different group of cells, and they will give rise to A peptide chain and B peptide chain. And in the lab condition, they will introduce disulfide bonds so as to combine both the chains. And now what you get is mature insulin, that is humulin. Option A says disulfide bond. Yes, that is absolutely right. Option B says intrahydrogen bonds, not no intrahydrogen bond, because you see whenever there is a structural conformation change, you would say, or you can say that if you talk about proteins, initially they are present as primary chain, then they become secondary chain, then it will get converted to tertiary, tertiary structure, then quaternary structure. This folding of the chain or combination of the amino acid chain that happens by disulfide bond. Hence, that is no. So option C says covalent bonds, no, and option D says both A and B, no. So the correct answer is disulfide bonds. So let us conclude the answer. What is the option? The correct option is option A. That is disulfide. I hope you understood the explanation. Thank you.